expression and regulation of gene expression. Keep in mind that when we're talking about this, is that if you have a chance, I don't know, I don't think this worked, but I'll try it again. Going full screen to see if it works. Yeah, well, make sure you go into Connect. Make sure you go into Anatomy and Physiology Revealed, right? So then you can take a look at, number one, the pictures of bones if you don't have a lab manual. Again, right? I don't mandate anybody buy a lab manual. So when people say that they, want, for some reason, we want you guys to spend money on more books, that's completely false. I've always told every one of you guys, if you want a lab manual, buy a used one. So make sure that if you want a lab manual, you buy one that's cheaper. I don't care if it's used from a previous edition. It's only going to be used as a reference. So when you're looking at models of the bone, yeah, that's when it becomes important, right? But you don't need a brand new one. So what we do have when we're looking at Connect is the ability to watch the animations. So go to Anatomy and Physiology Reveal on your Connect link, and then you'll see animations for protein synthesis, for DNA replication. So let's talk about how do we regulate gene expression. Just as important to know how much insulin to make, we also need to know when to make it. What would happen if you are fasting? You haven't eaten in 12 hours, right? And let's say it's Ramadan and you are a practicing Muslim. And because Ramadan, you're not eating or drinking any food or any kind of drink at all from sunrise to sunset. So let's say it's about 5 o'clock in the afternoon, right before sunset, right? Your blood glucose is starting to really dip because it's been so long since you've had food and you've already broken apart your stores of glycogen, right? And now it's starting to dip. As it starts to dip, what would happen if your body starts to make insulin at that time, right? What happened is that the insulin then, if it's produced in an inappropriate time, it causes the glucose in your bloodstream. That glucose then goes into every cell of the body. If your glucose levels are already low, that inappropriate release of insulin will cause it to fall off. So then your blood glucose will be at 20 milligrams per deciliter, meaning that it would go so low that in some people it can cause them to have, right, some kind of hypoglycemic coma or seizures even. So the last thing we want to do is to produce an enzyme or hormone at the wrong time, right? So regulation of gene expression is very important in that way. So during the lifetime of cell, rate of protein synthesis varies depending on cell signals that reach the cell. So when we want to produce insulin is when there's high glucose. So high glucose concentration in our bloodstream is the signal to release insulin. When your blood glucose level starts to drop, right, you no longer have that positive high glucose variable driving protein synthesis. Because of that, your insulin level starts to drop as well. That's exactly what we want. All nucleated cells, except germ cells, have the full complement of DNA, right? The weird thing is this, during development, differentiation occurs. Some segments of DNA are turned off in some cells, while those segments remain on in other cells. Now, the crazy thing is the insulin-producing cells are called beta islet cells. They're found in the pancreas. And we're gonna see in the pancreas, another group of cells immediately next to it called alpha islet cells. They produce glucagon. Glucagon is a complete opposite of insulin. Insulin, when it's released, adds glucose receptors to the plasma membrane of all your cells. Because you have such high numbers of glucose receptors, glucose then enters your cell from the blood vessel. perfect, especially once you've eaten. Glucagon is the opposite. Glucagon is released when your blood glucose is low, when you're fasting. What it does is it prevents other cells, except your brain and the spinal cord. It prevents other cells from receiving glucose from the bloodstream, saving the glucose for the brain and spinal cord. So it's completely opposite of insulin. Yet these cells are incredibly close together. Why does one cluster of cell produce insulin and the other cluster next to it produce the opposite, glucagon? Is because of differentiation, right? The alpha islet cells are going to have the genes for glucagon turned on. The gene for insulin is silented. The opposite's true in the beta islet cells. What makes it amazing is that they're right next to each other. So somehow those signals for the beta islet cells to stay forming insulin, right, only went to the beta islet cells. The alpha islet cells get different instructions. So then instead of producing insulin, the alpha islet cells produces glucagon, and the gene for glucagon is activated in them. And they get the signal to just produce glucagon. All right. It's kind of crazy just how close they are, yet they produce completely opposite things. Cell life cycle, right? So that's protein synthesis. 2 parts, transcription, translation, right? And then from that point on, it's probably easier to figure out, all right, what happens in transcription? Write it down. What happens in translation? Write it down. Now that's going to be very different than what we're going to talk about next, which is DNA replication. So in a cell life cycle, we'll notice that this is a normal cell life cycle. You go through G1 phase, S phase, G2 phase, and then mitosis. Notice that these phases are of unequal lengths, meaning that the mitotic phase is very, very short compared to the S phase, right? The G1 phase is a little bit longer than the G2 phase, right? They're unequal length. So what happens? I just produce one cell. That cell undergoes a G1 phase. During the G1 phase, we start producing more metabolic products, meaning ATP, right? We get more and more DNA bases, so then we can form more nucleotide, right? Why? Because the next phase is called the S phase. The next phase is where we're gonna actually undergo DNA replication. In DNA replication, we read each side of a double strand and add matching bases and a matching strands to the side that we read. So we're adding more and more DNA, adding and doubling the DNA in the S phase. Once we've doubled the DNA, we go through the G2 phase, which gets us ready for mitosis. This is where we double the centrioles, right? Where we start forming little axons that kind of come up, right? So all that happens, and then we hit the mitotic phase, M phase, where we actually split the chromosomes that have been doubled up previously. Remember, by the time we're done with the S phase, when we're at G1, we had 2 sets of 23 chromosomes. All of them are single arm. So if we look at chromosome number 1 for mom, it has 1 arm. We look at chromosome number 1 from dad, 1 arm. That's true for all the chromosomes. By the time we're done with the S phase, if we look at chromosome number 1 for mom, it's going to have 2 arms. The 2 arms means 2 different chromosomes. It's got the genetic material for 2 chromosomes. But since they're attached, they only count as 1. If we look at chromosome 1 from dad, same thing, right? Double arm chromosomes. Then in a mitosis phase, we're going to start to split the chromosome, the double arm chromosomes. Goes through prophase, metaphase, anaphase, telophase, right? There is a mnemonic here, right? IP or IPA MATC. So in the IPA MATC, and I'll write that down since it's not on here. should match I for the interphase. Interphase is a combination of all three of these, G1, S, and G2. K for prophase, M for metaphase, A for anaphase, T for telophase, and C for cytokinesis. So this, right, 4 represent this MATC and P, right? So prophase, metaphase, anaphase, telophase. And then cytokinesis, where we actually break the cell up. So this mitosis phase is the aligning of the chromosomes in the correct position. So then we can split them in half. As we split them in half, each cell that we're going to form is going to have one chromosome from one parent, right? One set of 23 chromosomes from one parent. I pay MATC. I for interphase, P MATC for the phases of the cell cycle, the mitotic cycle. Now, before we talk about that, let's talk about replication of DNA. Keep in mind that we're doubling the DNA. Unlike protein synthesis, right? In protein synthesis, we're just taking a small strand, right, of the genome that codes for our gene and transcribing only that small area of the double-stranded DNA. In DNA replication, we're going to copy not just one of the chromosomes, but all of them. And we need to be as complete when we're copying and as accurate as we're copying. We have billions and billions of bases for each chromosome. We're going to have to make sure that we're going to copy those billions and add a second arm to it. So in terms of replication of DNA, the parent original strands become templates for the new strand to form on. Two identical DNA molecules are formed by something called semi-conservative replication. This occurs solely in the nucleus and occurs in the S phase of our normal cell cycle. What do we do here, right? Here, what we're going to do, this is our original DNA. We're going to have an enzyme called DNA helicase that splits, all right, that unwinds the double-stranded DNA. Remember, you have hydrogen bonds here really tightening up the two double strands. Well, we need an enzyme that unwinds it so that we can read each side. So what happens then, right? I'm going to have DNA polymerase. DNA polymerase then is going to read each side right here. So for every adenine, you put a thymine. For every, in yellow and green, right, a guanine, you put a cytosine, right? So and with DNA replication, you read every single base all the way until you're done reading each of the double strands. Now, as we base pair, we form a second strand on the parent strand. So here the parent strand is in purple. As we read each of the bases here and follow the law of complementary base pairing, we add a second strand that's now pink, right? Same thing on this side. Now, what we're going to see is when you take microbiology, you take higher level of molecular biology, right? They will talk about lagging strands and leading strands. They'll talk about Okazaki fragments and ligases. For our class, we just don't have as much time to discuss it. But understand that when you do get into a class that's heavy and discusses, biochemistry pretty heavily, that they will talk about how we read one strand. And as we read one of the parent strands, one of the strands is going to be easy to read. And you're going to have one strand going a little bit faster. That's a leading strand. And it, basically is due to the fact that DNA polymerase, the enzyme that allows us to make that second strand, that new strand, that enzyme can only go in one direction. That's why you have a leading strand as it follows that law where it only reads in one direction. from 5' to 3'. Then you have a second strand that has to be formed reading the second old template strand. But as you read this strand, it's difficult because it's going in the opposite direction that it should. So even though you are eventually going to add a new strand to it, right? When it goes in the opposite direction, it lags behind, and this is called a lagging strand. Eventually, though, both original template strands in purple has a pink new strand attached to it. When this happens, I've doubled the DNA, right? This was one double-stranded DNA. Now I have one double strand to the left, another double strand to the right. Two double strands instead of the original one. Now, what we're going to see is they will be bound together, these two strands, these two double strands of DNA, bound together by a protein called a centrosome. Once they are bound together, it only counts as one chromosome, even though we have the genetic material for two. Once we split them apart, they count as two separate chromosomes. But when they're bound together with the centrosome, it only counts as one. All right, so after DNA replication, we have double the amount of DNA, but our ploidy number doesn't change. It will go from 2n ploidy number, and then after we should have 4n ploidy number. We don't, right? They're attached to each other. It still counts as 2n ploidy number. Right? Here's that IPMATC. Again, when you see double arm chromosomes, each of the chromosomes right here, remember, right? You have the centrosome right here, right? You have one chromosome on the right, one on the left. There's a protein connecting it. It only counts as one chromosome, right? Now, we do call these slightly different, right? We have a different name for each one. Both are called one chromosome. Each of these are called sister chromatids. This chromatid is identical to this one. The weird thing is, once we detach, see how these are detached? They're no longer called sister chromatids anymore. They're called chromosomes again, right? So just keep in mind that the semantics are very, very difficult. They're very confusing, right? When you have two double arm chromosomes attached at the protein centromere here, it's called this is just one chromosome, with each of the arms called a sister chromatid. But once we detach the chromosome, they count as separate chromosomes now. I know, I don't know why they call it that way, but they do. Right? Now, once we've doubled the DNA, how do we align it in such a way that we can form two cells out of the original one cell, right? And those two cells have or should have identical DNA to each other and to the parent cell, right? So once we have this parent cell right here, parent cell is going to undergo the cell cycle. When we're looking at a cell in the interphase, whether it's G1, S phase, G2 phase, right? Any cell that's in the interphase region, you will not be able to see the chromosomes. It is only when we are done with DNA replication that we're done with the G2 phase that we start prophase. Once we start prophase, then the chromosomes condense, and now we start seeing them, right? Now, what we're going to do is for every red chromosome, right? These are mom's chromosomes. In yellow, you have dad's chromosomes, right? We just arbitrarily giving it a little bit of sex here, right? So you have those chromosomes that are double arm, meaning they've undergone DNA replication. All right, now in the prophase, we start to see it. Remember, in mitosis, we need to produce two daughter cells from one original parent cell. By the way, right, you can imagine every single cell, the trillions of cells in our body are produced from the original fertilized egg. So all of our cells are produced from that original fertilized egg, which is crazy, right? So when the fertilized egg undergoes mitosis, it forms more cells. One cell becomes two, two cells becomes four, four becomes eight. You can imagine how many rounds of cell division before we have enough cells to form even a small, small baby. All right, so here you take a look, you see the chromosomes, and you see that they're double armed. What does this tell us? That we are at the very start of our mitosis phase. Very start is called the prophase. In prophase, these centrioles and the astral fibers starts to move apart as well. Right? We start seeing them and they're going to start moving apart. As they start moving apart, they're going to start to align. So these astral fibers are going to connect to the centromere. As the astral fibers connect to the centromere, they're going to move the chromosomes. So then all the chromosomes are found right in the middle. As it's all the chromosomes are found in the middle, right here, it's probably the better picture. When all the chromosomes are aligned right down the middle of the cell, we say that the cell is in the metaphase. Metaphase, midline. All of your cells, right, all of your chromosomes are down the middle right here. Why? Well, it's because this is what we want to do, how we want to orientate the chromosomes, so then we can split. Remember, we should have two sets of 23 chromosomes for mom. So she has one chromosome number one chromosome number two, one chromosome number three, one X chromosome. That's it, right? You have 23 chromosomes for mom. The other 23 chromosomes should be from dad. One chromosome one chromosome two, right? When we add it together, now we have one chromosome one from dad, other one from mom. Same thing with chromosome two, one from mom, one from dad. But how do we line it up so then we get that? Right? Simple. All right, here is mom's chromosome number one. One original, because it's double arm, we know there's a copy. Here's chromosome number two from mom. One original, and since they have a second arm, a direct copy. Same thing with dad. One chromosome number one, another copy. One chromosome number two, and another copy from dad. Since they're all lined down the middle, what we're going to do is split in anaphase, split the chromosomes in half. What happens if we split it in half right here? Here's dad's chromosome number one right here, and here's a copy on the other side. When we split it in half in anaphase, right, this arm goes over here. The other arm goes to the other side. As soon as we split the chromosome, the chromosome arm, as soon as we split it, they become separate chromosomes, remember? So this chromosome number one goes to this side. The other chromosome number one goes to the other side. So this cell eventually will have one copy of chromosome one from dad. This cell will have one copy of chromosome number one from dad. Perfect. We should have that. How about mom's chromosome number one? As we split it, one arm becomes a chromosome, right? So this cell, as it kind of moves this chromosome this way, well, this cell will get one copy of chromosome one from mom. Perfect, because the other copy is from dad. The other cell will get the other copy of chromosome one. Chromosome two, same thing. Lines right down the middle, and then we split it in half. As we split it in half, this cell over here will eventually get this copy of the chromosome number two from mom. The other copy of chromosome number two moves to the other side and becomes part of the other cell. So now it's got one copy of chromosome one from mom, one copy of chromosome one from dad, one copy of chromosome two from mom, and then the other copy of chromosome number two from dad, right? Perfect, which is exactly what we want. We want to split these chromosomes in half. As we split them in half, each cell will get one copy of one chromosome from one parent, right? It's not like this chromosome number one goes to this side. This copy of chromosome number 1 goes to the other side, right? If that happens, then this cell will get 2 copies of chromosome number 1s, but only from dad. This cell will get 2 copies of chromosome number 1, but only from mom, right? That doesn't match. So that's not good, right? So we don't see that. Right? What we do see instead is that all 46 chromosomes are lined up right in the middle, right? Chromosome number 1 from dad, right on top of chromosome number 1 from mom, chromosome number 2 from mom, and then chromosome number 2 from dad. Chromosome number 3 will be over here from mom. 3 from dad will be right next to it. All right, because of the way they're aligned, when we separate the chromosomes in anaphase, as we split it, each cell will get 1 copy of 1 chromosome from 1 parent. So mom will give you 23 chromosomes, dad will give you the other set of 23. Same with this cell. 1 copy of chromosome, 23 chromosomes from dad, 1 copy of 23 chromosomes from mom, right? They only show 2 chromosomes, but in your head, you can imagine that there's 23 chromosomes here, not just 2. There'll be 23 chromosomes here, right? From dad, not just 2. 23 from mom, not just 2, right? So in anaphase, we split the chromosomes apart. In telophase, we're then going to, right? We're then going to kind of start forming and start breaking apart the cell. In cytokinesis, we separate the cell completely. Now we have 2 different cells. These 2 cells should be identical to each other. As long as there's no mutation in the DNA, as long as we don't make a mistake when we undergo DNA replication, right? If there's no mistake, these 2 cells should be identical. Those 2 cells should also be identical to the original parent cell. So in terms of cell division and mitosis, this is how we reproduce cells, right? You can imagine, right? If you have 1 fertilized egg, you can't bring extra cells from mom and add to it, right? Those cells don't match that fertilized egg. So in order to make the baby, that 1 fertilized egg undergoes cell division. It gets larger. It undergoes mitosis, and then it splits in half. As it splits in half, you have 2 cells from 1. Those 2 cells are identical to each other. They're identical to the original parent strand. Perfect. Then 2 cells are undergoing mitosis, becomes 4. 4 becomes 8. All right, you can see how long it would take before we have enough cells to form a person. All right, next. Whoops. Right, let me see. All right, that's it, I think, for chapter 3. Right, now there's a lot of biochemistry and microbiology in the first couple chapters. The good news is after this chapter, there really isn't much more of this, right? So just keep that in mind. You don't have to tell me if you've emailed the answer, right? I'll look for everybody's answer. So just keep in mind, right? When we're looking at this, in terms of the big picture, the test is going to be very stringent, right? 50 questions. You're going to need to know, you need to study for it, or even starting now, even though the test won't be released until next Wednesday. So it will be released next Wednesday afternoon. It'll be due next Saturday at midnight. So I'm telling you now because I want you to plan ahead, right? It's not on our weekly to-do list because it's not, the test isn't released this week, but it is next week, and you need to start preparing now. Right, so let me finish up with that's it for chapter 3. We have a little bit more time, so I'm just going to go straight into chapter 4. So then we can at least start it off, and you can see what I expect from you guys. Whoops, I didn't need that. I'm going to